Right then, it's just gone 4 o'clock. So welcome to another of the SimSip webinar series, scientific webinar series. Today we're going to be hearing from Venkatesh from AstraZeneca, and his topic is looking at a recent paper that he's published in the British Journal of Clinical Pharmacology. And it's looking at pharmacokinetics under the COVID-19 storm. While he's going to be doing his presentation, there's opportunity obviously to ask questions. I think most of you, I think by default you're muted actually when you join, but there is a chat function. So please put your questions in there, and what will happen is either we can ask those questions as Venkatesh goes along, or if they come more towards the end, we'll save them for the end, and either I'll read them out or admin, who's in charge of IT here, who will unmute you and you can ask your question. So just to introduce you to Venkatesh, I'm sure most of you are very familiar with Venkatesh, and he's a very busy man, and he wears lots of different hats. So he's attained his PhD from Netherlands, and even from the start, as you can see, he's got a very collaborative nature in terms of working with Pfizer, Janssen, and Merck. So even in AZ now, he is highly collaborative, as you can see. He's working in lots of different areas, supporting PBPK, working in the area of PK/PD, and also he's quite a champion for PBPK modeling, and has published a lot of papers in this field. Talking more about his kind of collaborative side, he's deputy topic lead for ICH and 12 DDI guidance. Also working in the IQ, PBPK induction group. He didn't put on here either about his ISSX modeling and simulation group. There's probably not enough room for all the different things that you're involved in. But yeah, at the bottom as well, he's a big champion in terms of SimSip, and he's been chairing the consortium members discussion group as well for 2 years. So yeah, Venkatesh, we're really excited to hear your talk on your recent paper. So I'll leave it over to you.
Thank you, Oli. I just want to make sure you can hear me okay and you can see my desktop.
Yep, okay. That's all fine.
Yeah, thank you, Oli, for introducing me. Again, it's a collaborative work, as Oli mentioned. The work which I'm going to present today is collaborated with Certera, Manchester University, and also within AstraZeneca. It's a collaborative work between DMPK and clinical pharmacology and quantitative pharmacology. I'm not sure how many of you know, I moved from oncology DMPK to clinical pharmacology and quantitative pharmacology. I'm sitting in neuroscience, microbial sciences, and clinical immunogenicity group at the moment, and supporting some of the COVID activities. And this work was published back, submitted to the publication back in June 2020. And all the work which was done was before that. And there was a lot of additional data has been generated, which is not part of these publications. And it's also a very proud moment to contribute using physiology-based pharmacokinetic modeling for this pandemic COVID-19 to understand what are the gaps in the field of clinical pharmacology, whether we could use PBPK modeling to inform those gaps. That is the kind of a story which I'm going to present today. Disclaimer, again, this is a collaborative work between co-authors, which you can see here, bottom of the slide. And the views are coming from me and co-authors, and it should not be constructed as a representation of AstraZeneca. outline, I'm going to introduce COVID-19. Again, when I joined, I'm not an immunologist. I learned a lot from last 1.5 month time. I'll introduce COVID-19 situation and how the virology works and what are the treatment option, what we have at the moment, the challenges and opportunities, what we had over the last year for repurposing some of the existing drugs, and also show you the utility of quantitative modeling and simulation to understand the cytokine storm. I will get into the cytokine storm in a minute, why it is important for disposition of small molecules, and how we use PBPK modeling to understand that the reduced CYP3A4 expression because of cytokine, and how we could use PBPK, especially SIMSEP, for monoclonal antibodies, and also, if permits, vaccines. And finally, a few take-home messages. A bit of a COVID-19 refresher. So what you see here is a SARS-CoV-2 virus. So it needs ACE2 receptor, which is abundantly present in the epithelial cells. And what you see, this spikes is called S protein. And you have a ACE2 receptor. So there's a receptor binding domain, it's called. So when virus latches to the ACE2 through this receptor binding domain, it will get activated, some of the protease enzymes which are present in the cytoplasm, and it makes the virus get into the cell. Once it gets into the cell, it uses host cell machinery to replicate and also to assemble the parts which is required. For example, it will makes its RNA replication, and it makes its own the proteins which are required to express that protein, spike protein, and it will assemble back. It uses Golgi and epithelial reticulum machinery of the host cell, and then it will multiply. As a result of this, in the normal healthy respiratory system, so you do have alveoli, and you have the blood vessels here, and you have a diffusion of oxygen and carbon dioxide exchange constantly happening. Whereas in ARDS, which is acute respiratory distress syndrome, it could be a moderate damage or it could be a severe damage. If you look at the severe damage, the exchange of oxygen between alveoli and the blood is drastically reduced, because once the inflammation happens, what happens is that there will be a vasodilation, and also a lot of T cells activation happens, and the cytokines are released. Once the cytokines are released, there will be a lot of kind of a vasodilation, and the cytokine storm is happening there, and it attracts a lot of proteins, and it will make the inflammation worse. And also you can see the accumulation of fluids happening there. As a result of that, you can't really breathe, take oxygen enough, and exchange between the blood. As a result of that, the patient will experience a severe respiratory distress. I have a video. The idea of showing this video is not promoting AstraZeneca or anything, but just to get the basics of immunology, I asked Adam to play this YouTube, which is about 3 minutes. I hope you do enjoy it.
Okay, thank you. Give me 1 sec.
SARS-CoV-2, the virus that causes COVID-19, has distinct club-like spike proteins protruding from its surface. These spikes latch onto cells in the body by binding to receptors on the surface. Once attached, the virus fuses with the cell, and the viral genetic material passes into the cell, where it is copied, producing more viral particles and causing more infection. Scientific research is underway to combat COVID-19 in different ways. Firstly, medicines that may treat the infection by stopping the virus from replicating, by suppressing the body's overactive immune response, or protecting it from serious complications such as organ failure. Secondly, novel coronavirus neutralizing antibodies that can bind to the virus and stop it from entering cells as a possible treatment to prevent or lessen the impact of COVID-19 disease. Lastly, a vaccine as a preventative approach. There are multiple approaches to vaccine development to prime the body's immune response to COVID-19, using weakened viruses or viral proteins, or creating viral vectors with specific viral genetic code, or using specific viral genetic code, either DNA or RNA directly. Viral vectors are engineered to prevent them from being able to replicate and cause disease, but can carry specific genetic material to deliver inside a cell. The vaccine is introduced into the body through injection. Encompassing the genetic material of the SARS-CoV-2 spike protein, the viral vector binds to the cell surface and fuses with the cell. Inside the cell, the genetic material is processed to form the spike protein, which is expelled out of the cell and into the body. B-cell and T-cell lymphocytes recognize the spike protein as foreign and start to multiply to form an army of identical B-cells and T-cells. B-cells release antibodies into the circulation to bind to the virus and to neutralize it. T-cells help activate B-cells or eliminate infected cells. Both B-cells and T-cells form memory cells that are ready for when the body is exposed to the virus at a later stage. Upon exposure to the live SARS-CoV-2 virus, the immune response is primed to neutralize the virus and reduce the impact of disease.
Thank you, Adam. Do I need to reshare my desktop?
I've just given you the rights.
Okay. Okay. Thank you for that. Hope you got a little bit of the glimpse of what's happening in terms of the COVID-19 immunology and also the hope for treatment like vaccines. Of course, vaccines are gold standard, but it requires a lot of time. And I think you've seen the news, so we got 3 vaccinations approved last month. But before that, you can see in this slide, the world really marks 2 million coronavirus deaths worldwide. This is the death total number of deaths as of yesterday. You can see the larger the bubble, higher the deaths are. It's really alarming. And in addition to that, we also have this new variants coming up, like the South African, and the Brazil and the UK one as well. And hopefully these vaccines also will be effective for these variants. And yeah, this is just to show you the coronavirus deaths and how naughty is this coronavirus pandemic. As a result of that, again, last year, the necessity was the mother of invention. What you see in this slide is number of clinical trials happening worldwide. So this is as of yesterday. There are more than 2,500 clinical trials happening, which includes small molecules, monoclonal antibodies, vaccines, stem cell research, and many more, and plasma therapy, and also including the herbal medicines. So it's a massive, massive amount of work happening across the world. Of course, we need different platforms to combat COVID-19. In this slide, what you see is different options one can choose, because as a patient or as a subject, you wish to make your own decision. it could start with a plasma therapy, which is nothing but the convalescent plasma is harvested from the patient who recovered from coronavirus disease. And once you harvest that plasma, and they will have neutralizing antibodies, and that will be injected to the another person to protect from coronavirus. Yeah, it has its own success rate, but there's a lot of literature coming in. It is a kind of a mixed bag. And the second one is repurposed drugs and also new drugs. The repurposed drugs are quite straightforward. We were looking into antivirals, for example, remdesivir, which is a protease or protease inhibitor, and also baricitinib, which is a JAK2 inhibitor, and dexamethasone is the Il6 inhibitor or anti-inflammatory drugs. There are many drugs where we have clinical data already. We kind of repositioned that for coronavirus. And these three are already kind of approved emergency use authorization from FDA and other regulatory authorities. And the third one is monoclonal antibodies. So there are 3 monoclonal antibodies in pipeline. The 2 are approved quite recently. So these monoclonal antibodies is kind of a neutralizing antibody. So they latch onto the spike protein of the virus and stops binding to the ACE2 receptors. That's how it works. And their Lilly compound and Regeneron compound has been already approved, and also AZD7442, which is in phase 3 clinical trial. And the final one, the vaccine programs. As you know, many of people, we have mRNA vaccine and adenovirus vector vaccine from AstraZeneca. At the moment, AstraZeneca has been approved, not in US, but other regions. Moderna and Pfizer vaccine has been approved. We all know vaccines are gold standard to protect the pandemic, but there are many instances patients like immunocompromised patients may not able to take vaccines, and transplant patients, and also those who are allergic to vaccines can't take. And also there is another one, the feature is the antibody dependent enhancement. So it will, like for example, the vaccine will attach to the virus, but this vaccine produces more neutralizing antibodies that will have an FC region. That's possible that FC region can bind to the macrophages, and this once macrophages takes this virus with the neutralizing antibody produced by vaccines, still it could be lethal. There will be kind of immune toxic human toxicity could happen. So that is called ADE, antibody dependent enhancement can happen. And for treatment, definitely we need repurposed drugs, both new and repurposed drugs, small and monoclonal antibodies, to overcome the limitations of vaccine.
Venkatesh, I think you've got a little box in the top right-hand corner of the presentation. If you could just move that out of the way.
Right, sorry.
Yeah, thank you.
Okay. Yeah, this little cartoon, what you see here is number of compounds which are repurposed. As you said, as I said, repurposed drugs are quite first step to combat this COVID-19. Of course, new drugs also can be developed, but it is time consuming. What I would really like your attention is to, we had around 3 or 4 monoclonal antibodies in the pipeline last year. From Roche, from many other companies, and also small molecules, including remdesivir, dexamethasone, and many other antivirals. So you can see it's like many small molecules do act at different steps of COVID-19 infection. For example, Camastat inhibit this trip RSS2, which is required to disintegrate S1 and S2 protein of a virus so that it can latch into. And also, for example, dexamethasone also acts as an anti-inflammatory, which inhibit the cytokines IL6. There are many drugs which were repurposed, including AstraZeneca, acalabrutinib and ibrutinib, which is the BTK inhibitors, which appears to reduce the cytokine levels. And if you look at, for example, remdesivir clinical trial study here, it was published back in May. one attention I would like to draw your attention is the diversity of inclusion criteria. For example, usually for a typical new drug, we tend to recruit patients from 18 to maybe 55 years of age. But in this case, these are like started with the healthy volunteers, or starting from 18 to almost 65 plus years. And you can see the distribution, the max, I think there was a 85 years old patient also was there enrolled in this study. And also we had comorbidities, the subjects who had comorbidity, for example, here, liver disease and chronic kidney disease, and also obesity and cancer, is a kind of a mixed inclusion criteria. There was no strict inclusion criteria was followed. The basic thing was mainly here is efficacy to combat this COVID-19. A point I'm trying to make here is, for example, remdesivir, we didn't have a dedicated clinical studies to understand the comorbidity, intrinsic and extrinsic factors. So it kind of like came as a storm, and the clinical trials included who were getting into the hospital just get protection, or if possible to save that patient, the remdesivir was given. So this is the kind of data set we expect for many other compounds. What you see here in the heat map, what we have listed here, number of compounds, repurposed compounds, and on the first row what you see is what kind of data is available. So many new drugs, for example, ibrutinib or acalabrutinib, we had quite a lot of data. It was extensively studied. Ibrutinib is extensively studied, and acalabrutinib maybe is also extensively studied, but still there's some gaps. For example, renal impairment and hepatic impairment study data was not available at that time for acalabrutinib. And if you look at the remdesivir, there's only the single ascending dose was available at that time, and there are a lot of gaps involved. You can see all these thread boxes there. Idea of using the modeling and simulation or quantitative pharmacology, whether we can fill in if the verified and validated PBPK models are available. That is the kind of intention we had. We looked into all the compounds where the verified and validated and submitted to the regulators PBPK models we gathered together. And then we looked into what are the gaps exist for many compounds which are repurposed for COVID-19. And then we run the simulations. It's not only that, and also we summarized all the ADME, disposition process, adverse events, and many more aspects in that paper. And this is a nice review article came from Associate Editor of CPT and Amin, which was published in CPT back in May 2020. So it's again, it's clearly showing how we could use quantitative pharmacology methodology, either PBPK or POPPK or exposure response relationship, QSP, many more, and how we can establish benefit risk relationship for a given compound, repurposed compound, and also how to mitigate the impact of trial disruption. So it's a nice review article I recommend to go through. So it's clearly highlighted here that quantitative pharmacology methodology, including PBPK and PKPD modeling, plays an important role here. I'm coming to the cytokine storm. This is a nice cartoon showing you here, for example, the first upper circle shows SARS-CoV-2. When a patient gets exposed to this, kind of like 40 to 50% of the patient, they combat the COVID-19 infection, but 20 to 30% of the patients will have a hyperimmunity reaction. As a result of that, hyperimmunity immunoinflammation is because of the activated NK cells, natural killer cells, CD4 and CD8 cells that will secrete TNF alpha and IL6. And these are the kind of cytokines, cytokine, the mediators, which will causes this the damage by kind of making all this inflammation worse. So that's why it's called kind of a cytokine storm. So cytokine storm can be also mediated by like kind of many monoclonal antibodies, like especially the bispecific antibodies through CD19 or CD22, they appear to, if you have a target on CD19 or CD22, it appears to have also a cytokine distress syndrome. So there are many ways you can get this cytokine storm, but SARS-CoV-2 also causes this kind of cytokine storm. And the point I'm trying to make here is, like, as I mentioned, those patients who have cytokine storm had high mortality. In this paper, it was nicely mentioned. And also, you can see around 50 to 75% of the patients who were hospitalized, and many patients also died. They all had this COVID storm, whereas around only 20 to 25% had no storm COVID-19, but still they were in hospital, but it was not as fatal as those patients who had this COVID storm. And why this COVID cytokine storm is important for pharmacokinetics. This is a nice article showing you what happens with the cytokine storm. For example, rheumatoid arthritis, which is an autoimmune disease, or cirrhosis, or in even cancer. We know that presence of tumor and rheumatoid arthritis, it enhances release interleukin-6. So once it is released, it gets into the liver, and it will decrease the mRNA expression. For example, CYP3A4. There are more than 5 to 6 papers. There is a clinical evidence, and also the in vitro and preclinical model established to say that IL6 reduces mRNA, CYP3A mRNA, not only CYP3A4, many other CYPs, and thereby the expression or abundance of CYP3A4 in the liver and the gut, and also maybe in the kidney, reduces. And many of small molecules, for example, acalabrutinib or ibuprofen, they are mainly metabolized by CYP3A4. They are quite sensitive CYP3A4 substrate. If you have reduced CYP3A4 content in the gut and the liver, you might experience higher exposure. That's the point I'm trying to make with this cartoon. And what we have done here is systematically collected the IL6 levels. What you see this in the bar plot is healthy, you have around 20 picogram per ml. In cirrhosis and rheumatoid arthritis, you see it goes up maybe 6 to 7-fold. And also cancer, there is evidence that 4-fold. We looked into all the papers published at that time, sometime in May. And also recent papers show that the COVID-19, the IL6 can go up to all the way 500 picogram per ml. But at that time, what we had was around 100, which is close to the cancer. And many, those who are working in cancer also have noticed midazolam clearance reduces in the cancer patients, and as a result, you do see 2-fold higher exposure. So there is evidence that cancer and many other rheumatoid arthritis patients tend to have higher exposure because of the reduced CYP3A4 expression. So there is an association of IL6 levels and CYP3A4 expression. So that was our intention. So what we did in this paper was we collected all the data, ADME data, DDI, adverse events, and efficacy data where possible for these 15 repurposed drugs, both small molecules and large molecules. And the idea was kind of, this is a themed issue. The idea was to provide all the necessary information that is related to pharmacokinetics and clinical pharmacology. That's what we did. Yeah, that's all. I think, like, if you're interested, please go and see this paper. I don't know how many of you have gone through this paper already, but I'll briefly touch upon the highlights of this paper. So as I mentioned, there were a lot of gaps, and the heat map was shown here. The objectives of this themed issue or themed paper was mainly to summarize and inform the clinical pharmacology aspects so that physician or those who are treating these COVID patients can look into this ADME if necessary. We summarized ADME, PK/PD, DDI aspects of it, and also looked into the effect of intrinsic and extrinsic factors like renal impairment and effect of the extrinsic factors, and we tabulated that. And also we looked at the in vitro IC50s or IC90 values, to see whether the unbound plasma exposure is achieved above the in vitro IC50 or IC90 for the SARS-CoV-2 cell lines. At that time, we had very limited data in terms of IC50 or IC90, but we know from FDA paper that we should be looking at unbound IC50 values and also unbound plasma concentrations and also unbound target site concentration. So we made it very clear at that time. And we looked at all this information, the in vitro PD data, and we tried to predict the target site concentration because virus replicates in epithelial lining fluid and the upper respiratory tract, and that is kind of target site we need to hit hard. So we gathered all this information and we summarized in this. And we used PBPK modeling. We used SimSip as well, and also for stakeholders, we also applied AstraZeneca PBPK tool to make sure we get the reproducible results between SimSip and other PBPK tool. We did that as well, and finally we were able to publish it. So this slide shows some of the intrinsic factors like age, race on the drug disposition. So what you study here is on the left-hand side is the effect of age. Since many patients were 65 to 98 years of age, geriatric patients. So what we did was, I'm not showing all the compounds plots here, but I'm just selected 4 compounds just to show you. And the if I think maybe I should take the dexamethasone here. So what you see is the shaded area and the black line is for the healthy volunteers. And then the shaded area is the prediction intervals. And then the green is 40 to 65 years of age. And then the red one is geriatric patients. So as you expect, the expression of the enzymes reduces with age. So dexamethasone exposure was going slightly higher than the prediction intervals of the healthy volunteers. Yes, but dexamethasone is well tolerated, and this twofold higher exposure is okay. But some anti-cancer drugs like acalabrutinib, ibrutinib, and baxitinib, and many others, not cytotoxic, but targeted kinase inhibitors might have the safety issues. So we need to really worry about what is the exposure changes, and we need to understand the exposure response relationship to make decision of the dose. For example, ibrutinib, even though it is approved 560 milligrams QD, but in their trials, they reduce it to 460, if I'm not mistaken, and many other compounds, they tested a slightly lower dose than the approved doses for this purpose. So that is a kind of point which I'm trying to make here. And also on the right-hand side, what you see is the effect of the race, Japanese versus Chinese. We know that there is expression differences and also demographic differences between Chinese versus the Caucasian. So we also looked into that and we reported that here. And also very important to study organ impairment. So as I shown in one of the remdesivir trial design, there were a lot of patients enrolled who had comorbidities. We were interested to see what happens with this repurposed drugs is given to hepatic impairment and hepatic renal impairment. I know IQ paper has came out last month for the hepatic impairment, and still we have some limitations of using SimSip for predicting hepatic impairment. But this is the science evolving, and what we had at that time we used. On the left-hand side, what you see is hepatic impairment, and on the right-hand side, what you see is the renal impairment. Yes, for I think for azatinib, there is slightly higher exposure, and also for dexamethasone. This is the kind of information you get, and then you can speak to your clinical team within a company to see whether the dose adjustment is required or not. This is really useful information to gather and provide to clinicians and for the trial design decision makers, so that they have this information when go for a clinical trials. And this is the sensitivity analysis we did. Actually, what you see on the left-hand side is for acalabrutinib, and then the right-hand side is for the ibrutinib. So you know ibrutinib, there's a gut metabolism involved for ibrutinib, FGF 0.5 if I'm not mistaken. The idea is, for example, IL6 was increased by 5-fold. We know IL6 reduces CYP3A4. So what Iman, one of the co-author, did was a kind of reducing CYP3A4 abundance both in the gut and in the liver, and look at the AUC ratio, how much is going there. So if you reduce by, say, the gut to 0, or maybe 18, 18 picomole per the whatever units in the gut and in the liver, by like say 3-fold or 4-fold, the maximum AUC fold increase for acalabrutinib is around 4-fold. If you compare with the ibrutinib, as it is very sensitive and also the gut metabolism is involved, the AUC ratio change when you reduce by maybe 50% or both in the liver and in the gut, you tend to see higher AUC ratio changes in the patient. So we need to be a bit careful, especially when you're dosing a patient with renal impairment and having a cancer as a comorbidity, there might be higher exposure we might expect. But in reality, we don't have this data. For example, in COVID-19, whether actual CYP3 expression, whether it reduces or not, but it's an hypothetical assumption that IL6 is increased, and eventually it will reduce the CYP3A4 expression. That is our assumption. And again, the beauty of having sensitivity analysis in the SIMSIP, we can pressure test all these different scenarios. But, oops. Hello, can you hear me okay?
Yeah, we can hear you.
Oh, okay, sorry, I heard some beep. Okay, so we have done this sensitivity analysis and we provided this for the 4 drugs, and just to see which one really we need to take, give some great attention in case we might see higher. And the next step was to simulate the target site concentration. So what we did was we used the SIMSIP again. I don't know how many of you are familiar with the permeability limited lung model. So you have different compartments within the lung. So you have blood and you have lung mass. You have an epithelial lining fluid and the alveoli. And you have for the right lung, left lung, and the lower airway and upper airway. So it's like nicely compartmentalized, and it takes into account of the actual disposition of a compound from blood to all the way to alveoli. So what we did was we used all the validated verified compound files, and we simulated, we predicted the epithelial line fluid concentrations for the small molecules. One example which provided here is for acalabrutinib unbound epithelial lining fluid concentration over the time. Acalabrutinib has a short half-life, and it is lung and blood. Yeah, the lung is highly perfused drug, and you expect to see the plasma exposure mirrors with the lung exposure, which was not a surprise for us. So I think SIMSIP, the permeability limited lung model, nicely allows to simulate it, and we are not the first person to implement this, but many papers have been implemented and verified with, I think, anti-tuberculosis drugs and quinidine, I believe. Yeah, there's a lot of verification has been done, and we were able to also verify with RAT-KP data. We had some RAT-KP EU data. We were able to correlate with that as well. So this is the plot showing a correlation with the PD. PD here is the in vitro IC50 or IC90. So we know we need to hit hard because hit all the viruses which are remaining. That's why IC90 is a better metric than the IC50. And what you see here is the plots of different drugs. On y-axis, you have a lung unbound concentration, and then the dashed line is the target IC50 or IC90 concentration, and how long the exposure is above the target, for example, IC50 or IC90. That's what we were trying to achieve. I do appreciate that at that time we didn't have the IC90 values for relevant viral cell lines. We chose whatever data was available. I think today I was in a workshop on COVID where the institute from Australia has generated IC90 values using the relevant SARS-CoV cell lines, and they provided that IC90 values. So hopefully we will update this plot going forward for all the drugs, but this is as of May 2020. And this is a paper came from Sartara showing azithromycin, why it didn't work. This is a nice piece of work. So azithromycin is a monoprotic base, and monoprotic base, I'll come to that in a minute. The bases and high pKa value tend to accumulate in the lung because of the lysosomal trapping, and also because of the changes in the pH. What Karen Yu and colleagues have shown here is in the control, for example, there is no change, assume no changes in the pH happening in the epithelial cells, and is a normal lung. So what you see here is the green shaded area is the IC50 value of azithromycin versus this, the HIV, I believe. And then what you need to see is the yellow line, which is the epithelial lining fluid unbound. So azithromycin with approved therapeutic dose or regimen, we are not able to achieve that IC50, the shaded area there, the green shaded area. So I think that the dose what is being tested is suboptimal. And also there was a lot of papers back in March and May, people saying, oh yeah, chloroquine and azithromycin causes accumulation in the lung because they are monoprotic bases because of the lysosomal trapping. And what Sutter group has done, okay, if you change the pH, what happens, whether accumulation is going to happen or not. So yeah, we see some accumulation because of the changes of the pH and the lysosomal trapping, but still this ELF is not making the bar, reaching bar of this IC50. And also we know azithromycin is a renally eliminated drug, and if you reduce the GFR, whether that will make any difference there. So all three scenarios, azithromycin is still not reaching the required target concentration, suggesting that azithromycin may not work and correlate with the poor efficacy. And there are also a lot of papers came on chloroquine. There was some QTC side effects. This nicely brings the concept of the lysosomal trapping. So what happens is if a compound is a monoprotic base and has a high pKa, they tend to sequester in cells and makes the pH of the cell to more acidic. So what you can see here is on the right-hand side is 7.2 is normal pH or the blood pH, and when drug gets into the cell and it traps, because it's a monoprotic base, kind of brings the pH, alters pH, and it gets sequestered there. And as a result, you can expect the lung concentration to go up. That's what is shown here on the left-hand side plot. Lung plasma partition coefficient. This is normal pH, and then what you see is the gray line with the lysosomal pH of 5.5. You see the exposure going up at least tenfold. This is assumption. People at that time, 2020, chloroquine is yeah, that approved, the dose regimen is good enough, and it should give us efficacy, and that's where more than 300 studies with the chloroquine was initiated at that time, assuming that we have enough exposure reach in the target site and also in the plasma. But that not really translated well because the IC90 against this SARS-CoV-2 was very weak for chloroquine and many other reasons. And this is the common question we tend to get from regulators and also governance, how well one PBPK platform performs with others. This was asked within AstraZeneca as well. So what we did was we had an in-house lung PBPK model. We tested that and compared for one compound, dapagliflozin. So they were very comparable. So I don't have like-to-like plot here, but the point I'm trying to make, unbound concentration we reach in the lung is around 22 nanomolar, which matches with in-house PBPK model versus Simpson, which gives the confidence that yes, we are able to predict this ELF concentration reasonably well. And coming to this monoclonal antibodies, we would have liked really have this multi-compartment model for lung for monoclonal antibodies, but but unfortunately we didn't have this multi-compartment model structure as we have for the small molecules for the monoclonal antibodies, and we couldn't do much of PBPK model to predict the ELF concentration for the monoclonal antibody. However, we can output lung concentration as one compartment lung concentration, and you can see how much drug is reaching to the lung, but I'm not sure how much useful it is. But yeah, this is something, a lot of collaboration is happening between different pharma companies with Satara to build multi-compartment or lung model in monkey going forward. And this is something we could have a wish list. A pandemic is not this coronavirus situation is not going to end soon. It might come back again. It's good to have this kind of lung multi-compartment model for monoclonal antibodies and for beyond peptides and things like that. So this is just a point I want to make. I'll not go in detail of what is the structure of all those things. And you'll be curious as well. So we have vaccines now. Do you think SIMCIP can be used for vaccine development? Yes, there are efforts being happening. For example, this is the slide Pete Van der Graaf presented in one of the Satara webinar. And many of you may not be aware of this. Immunogenicity simulator within the QSP, it is interlinked between the SIMCIP biologic PBPK model and the immunoresponse model within the QCP. They talk each other. And I think, yes, there are a lot of inputs we can provide. For example, the heterogeneity in the patient population or population of interest, Monte Carlo simulation can be run using SIMCIP. You output that for the biologics and connect with the QSP model. That can be done. And it was shown such kind of interaction, interlink between SIMCIP and the QSP model within the this platform, QSP platform developed by Certara, has been implemented for the hepatitis B vaccine and also for the anti-drug antibody against adalimumab. And there were some simulations, but I'll not go into detail, but there is a potential for PBPK to be part of this QSP, as many of you know. With that, I think the conclusions are, we use this PBPK model using SIMCIP to study the intrinsic and extrinsic factors, and we reported that altered exposure and how much fold was different, and if there's any mitigation plans are required. And yeah, all clinical studies we can't study because of this time-constrained COVID-19 drug development situation. And the mechanistic lung model, again, most of them are simulations, but we were able to also compare with the in-house rat KPU data for many of compounds. And again, having some preclinical data, lung tissue concentration or PET imaging data would help to confirm what is the predicted or simulated lung concentration versus observed lung concentration. I think back in maybe a couple of years ago, we thought we'll use this PET data coming in for brain exposure, but that the collaboration didn't really completed yet. But this is something, if you have a lung PET data, we could use with a radius tracer of a compound, we could use that data to verify it. But at the moment, it's all simulations. Yeah, there's some verification was done for anti-tuberculosis drugs. And in the end, again, we need both vaccines and drugs, monoclonal antibodies and large molecules. And in my opinion, yes, PBPK plays an important role. I hope I've shown how much PBPK could be used to inform drug development. What I've shown here is mainly the ADME and the DDI side, but it could be also used for absorption side. For example, acalabrutinib was supposed to be dosed with IV dosing with mixing with the Coca-Cola. There was, I think some work was done. I'm not sure how much work has been done. Absorption side, and there was a beautiful work was done to some extent. I don't know the details, but yes, absorption PB, visually based biopharmaceutics modeling also can be used to inform the trial designs and make decisions. With that, acknowledgements, again, it's a collaboration between different functions within AstraZeneca, Oncology DMPK, CPKP, and also with Certara, and also with the Manchester University. I'll hand over to Oli, and if there's any questions, I'll happy to take.
Thanks, Venkatesh. That was really great. We do have a question. David Colton's put his hand up. I don't know what the question is, but maybe he could be unmuted and ask a question.
Yeah, I've just unmuted him.
How about now?
I can hear you now. Yeah, perfect.
Yeah.
Excellent. Thank you for the patience. Excellent talk. Question, when you were talking about the modeling of certain repurposed drugs, such as lopinavir, I wanted to make sure I heard correctly. Were you able to find, or were you not able to find instances where it looks like the inflammation due to COVID-19 itself had changed CYP expression and thus CYP-mediated clearance?
Yeah, so yeah, we were not able to find the CYP3A expression levels, but what we were able to find was the IL6
Okay.
levels in the serum. So that was really associated with CYP3A4 expression. So that is the assumption we made.
Excellent. Yeah, I would. This is a topic that overlaps one that I'm very interested in. I did see that August, September, October, each month there was a paper that came out that looked at pharmacokinetics of lopinavir or darunavir, CYP3A substrates in COVID-19, and in those patients they either had marked increases in IL6 or CRP or were very sick. So it all matches up exactly with what you're saying.
Right. Okay. That's good. I've not come across that paper. Thanks, David, for pointing out.
Sure. I'll email you after the talk.
Excellent. Thank you.
Okay, thanks. There's another question from Iman on mute.
Yeah, give me 1 second, please. Hey, man, I've unmuted you.
Hi, can you hear me?
Yeah.
I'm wondering if you combined all these factors and if you created a COVID patient population or not.
I couldn't hear your question, Iman. Can you repeat it a bit louder? Sorry.
Is it clearer?
Yeah. No, it's clear.
So I was these possible PK changes and you created a COVID patient population.
So PK changes with the COVID-19 treated patients with what, like any?
I think the question is basically whether you're able to create a specific COVID patient population.
Oh, right. Okay. Now, yeah, we mimic the patient population in the sense the demographics we tried to match. For example, if I think we did some simulations assuming if a patient has cancer as a comorbidity and obese, and we did not report in the paper, but we were playing around on that, changing some of the parameters and see how that will affect the exposure and things like that. So if your question is, okay, did we build kind of a COVID representative population? So yeah, we were trying to match demographics as much as possible and also into this comorbidities and things like that. But we did not have any PK in those COVID-19 patients. As you recognize, it's quite difficult to withdraw the blood and things like that. But I think what David was mentioning, maybe there is some papers, recently maybe there may be some papers might have come across, which I need to look into and see for compound. I especially remdesivir, at that time there was no PK was reported in COVID patient, but I saw a lot of papers coming out. But yes, this is to be done to answer your question, but that can be done as well.
Next we've got Neil Parrott. So if you just unmute.
Yeah, just unmuted.
Oh, great. Thanks. Hi, Venkatesh, and great presentation. And, you know, great work by AstraZeneca to get the vaccine out so quickly. So, you know, congratulations on that as well. My question is around the simulated unbound concentrations. So you showed slides of unbound lung concentrations, and you also showed, total lung concentrations or increasing KPs. And I'm wondering, usually we would assume that unbound concentrations are at equilibrium. So unbound plasma is often used as the surrogate unless we have mechanisms that are driving a gradient. So I'm wondering what is the value or what is behind those unbound lung concentrations in your slides?
Yeah, absolutely agree. So this is a big debate happened in ACCP with FDA colleagues and many others. What is the point of measuring the target site concentration when unbound plasma concentration equilibrate with anyway tissue concentrations? Yes, I see less of importance, but however, especially the drugs, for example, monoprotic bases like chloroquine and has this transporter liabilities and things like that. In the lung, we may not see the much transporters. Maybe I'm wrong, but yeah, for the compounds with the monoprotic base where lysosomal trapping is high, you might get some different picture. The unbound plasma concentration may not be really unbound concentration of the lung for monoprotic bases and some specific class of drugs. That was the intention for us to also simulate. But I agree with you, unbound plasma concentration correlating with IC90s is much relevant metric, and that has been shown that's much that is enough by FDA colleagues. I agree with you. It's again, we always talk about this target site concentration linking to the efficacy and adverse events, especially for the chloroquine. We don't know how much is accumulating in the heart because there was a QTc prolongation because of the chloroquine. Is it because just accumulation and might not have reflected with the unbound plasma concentration, or maybe blood to plasma ratio may be different for chloroquine? I think validating with the different metrics would be ideal. Good to know, and I agree with you. Site concentration measure is very difficult. A target site concentration is very difficult unless you have a PET study.
Thank you.
Yeah.
You have another question from Amin, I think. So I'll just unmute Amin.
It could be a question, it could be a point. So we'll see.
It is a point. Can you hear me?
Yeah.
It's going back to what Neil was saying. I'm not sure if Karen is online or not, but one thing on the unbound concentration, I think that we have to be careful with regard to the IC50 or EC50 or 90 or whatever that you are measuring, is that many people actually they are equating what they have measured in the in vitro system with what they are seeing in the in vivo, without correctly modeling the in vitro system. So while we agree with the FDA completely that the unbound is the one that is making the effect, etc., we do have problem with those people like all days. Neil, you remember from the MBI days that they were looking at, for instance, ecstasy, and then they were dropping the concentration in the tube, and they were assuming the concentration that is going to cause everything is the nominal concentration they have put in the system. That is wrong. Many of the IC50s or IC90s that people have measured, they have been on the basis of nominal concentration rather than actually seeing what is happening in the Petri dish or wherever they are measuring these IC50s. Therefore, we need to go back to all our in vitro systems and actually measure those IC50s properly with all the time variant that is happening to whatever we have put into the system, whether it is basically churned out, it is sticking to the tube, it is sticking to actually to components of the protein, etc. And therefore, the nominal concentration is not necessarily what we are actually putting there. So I wanted just to add that, as a clarification. While we agree with many of the things that FDA colleagues have said, we have got also points of disagreement with the way that the link between in vitro and in vivo actually works.
Thanks for that.
And
So,
you've got another question from Shola.
yeah, that's right.
Yeah, I'll just unmute her.
Hi there. Thank you so much for the presentation. I found it quite interesting. I was just going to ask for a bit of a more clarification on the point you made about the KP values you obtained from rats. So I wonder how did you use that value with respect to your simulation in the lungs to determine the concentration, the unbound concentration in the fluid in the lungs. So I don't know if that question makes any sense.
No, I think it makes. So we had KP values in the rat using this QW quantitative whole body imaging, if I'm not mistaken. So that data was available from our stock studies for some of the compounds. So we had a KP values there, and also luckily we also had for a couple of compounds we measured unbound concentration in that particular tissue, for example lung, and also the plasma protein binding. So we were able to derive a KPU in a crude way. And then in the KP, so in the SimSafe, again, we can output the KP and the KPUU values as well as we input compound fraction in the further lung tissue in the SimSafe simulator. So we can obtain. So we can compare KP, just a partition coefficient, without correcting for the plasma protein binding and the tissue binding.
Mm-hmm.
And also we can compare KPUU, which is makes more sense. So we were able to compare in a crude way. They were not so off in the sense, for example, if I am not mistaken, acalabrutinib KP was about 0.7, and in simulated and in the rat we got around 0.65 or something like that. It was in line, and also KPU was also within line. So based on that, I was saying, yeah, we were able to predict reasonably well after single dose PK. I hope I have answered your question.
Yeah, thank you.
Okay, thank you for that, and thank you again, Venkatesh, for your excellent presentation. So just before people go, I just wanted to highlight that next time we've got a talk from Sheila Peters from the IQ, so it segues quite nicely actually from what Venkatesh was talking about in terms of the organ impairment populations and also his link with the IQ. So what they're going to be looking at is the predictive performance in predictions for renal and hepatic impairment populations. And again, this is quite a collaborative effort, so it's across different pharma and different compounds. So we'll look at kind of where predictions are good, maybe some where they're not so good, and I guess it really drives us forward in terms of that research and getting these kind of databases together and realizing all the gaps. So yeah, it should be a really good presentation. It's a recent publication, so if you have a chance beforehand to read the publication, that's also quite a good idea. Thank you, everybody, and take care. Till next time.
Thank you.
Bye.